Hi everyone, I'm Brittany and I'm proposing a research project titled C-section versus vaginal delivery, the impact on intestinal microbiota and the pathogenesis of type 1 diabetes mellitus. It aims to answer the following question: what is the influence of the intestinal microbiota on the pathogenesis of type 1 diabetes mellitus, and does delivery method during birth influence this? Currently, the understanding of the pathogenesis of type 1 diabetes is that the disease is the result of the autoimmune destruction of the beta cells in the pancreas as a result of genetic and viral factors. The human microbiota is now thought to be involved due to the unexplained increase in cases recently in the developed world. The literature supports this idea with multiple murine studies showing that altering the microbiome has a direct effect on whether a mouse develops diabetes. Recent human studies have also shown that there is a significant difference in the gut microbiota between healthy children and those with diabetes. There's also a distinct change in the microbiota of predisposed individuals before and after the development of type 1 diabetes. An individual's microbiome is affected by exposures throughout life, one of the first being childbirth. There's a significant difference in the microbiota of those born naturally and via a C-section. This difference has been implicated in the development of some disease, such as asthma. There have been many preliminary studies that have looked at whether mode of delivery has an influence on the development of diabetes, but these have been inconclusive. This study has three clear aims: to confirm the changes to the microbiota in newly diagnosed diabetics, type 1, to study the mode of delivery of those presenting with new onset type 1 diabetes, and to characterise differences in the intestinal microbiota between natural-born diabetics and caesarean-born diabetics. It is hypothesized that there will be distinct differences between the microbiota of type 1 diabetics compared to non-diabetic first-degree family members, that there will be a high proportion of diabetics born via C-section, and that there will be a significant difference between the microbiota of diabetic children delivered through the vaginal canal compared with those delivered caesareanly. This study is a single-site study that will be conducted in conjunction with Perth Children's Hospital and the Children's Diabetes Centre based at the Telethon Kids Institute. Over 2 years, 125 participants will be recruited to the study. Patients will be recruited from inpatient wards and outpatient clinics. At presentation, patients will be consented, asked to complete a survey and a stool sample, analysed by 16S RNA sequencing. Family participants that live in the same household will be asked if they'd like to participate as part of the control group. Statistical analysis will be completed using SPSS using a variety of methods such as paired samples t-testing. This project will help to understand type 1 diabetes, one of the most common chronic childhood diseases. Currently, there are 130,000 people living in Australia with the disease and an estimated annual cost of $570 million. The seriousness of presentations, lack of curative treatment, and unexplained increase in prevalence indicate this is a problem that needs addressing. However, this is prevented by lack of understanding surrounding the pathogenesis of the disease. Understanding could lead to a creation of a therapeutic agent or a screening program that could highlight risk. Knowing the effective delivery method could help to stratify risk and highlight differences in potential interventions. This work would revolutionise the understanding of diabetes and provide hope for preventative or even curative measures.